So single molecules well separated in a field of view constitute essentially a perfect point optical source, and this results in many applications for the technology. So here is an example of using colored single molecule tags to measure the location of features on, for example, proteins to a resolution far better than the optical resolution limit. Remembering that these sources are really ideal point sources, when imaged by an optical system, they will form an image of finite width that is completely reproducible because it is simply a reflection of the performance of the optical system and not a reflection of what comes from the sample, because these are perfect point sources. Therefore, if you have two dyes, in this case a purple dye and a yellow dye, even although their fluorescence may be by the optical system considerably overlapped, since this point spread function is known, one can fit it and thereby determine a very small separation of these two peaks. Because the colors are different, one can use a color filter to measure each distribution separately, and the result is that a spatial resolution of the order of 10 nanometers is readily achieved. Very similar techniques let one track single particles that are moving. For example, membrane proteins in a lipid membrane move around, diffuse on the surface of cells, and so one would like to know what this diffusion is. So here is an image of the single molecule fluorescence from dye-tagged membrane proteins, and by simply mapping the position of the dye as a function of time, so here is an XY plot showing how the dye has moved in equal intervals of time, one's actually mapping out the step in a drunkard's walk, and one can do several things. First of all, one can deduce the diffusion constant from this, simply because after a number of steps, the end-to-end length will be related to the number of steps by this diffusion constant. Secondly, one can test the model of diffusion. Are these proteins truly diffusing randomly in 2 dimensions, or are there features inside the cell that act as tracks that tend to limit their diffusion? Here is a beautiful way of measuring dynamics in nanosystems, which is Foster resonance energy transfer. This relies on having 2 dyes in close proximity. Now, as shown on this scheme here, one of the dyes absorbs in the blue and emits in the green. The second dye absorbs in the yellow and emits in the red. So the idea here is that the fluorescence emission of the first dye overlaps in energy the absorption spectrum of the second dye. Now, if these 2 dyes are close to one another, then let's just look at this. Here is the more energetic dye, which then is getting ready to emit fluorescently here. This excited state here constitutes a dipole. The second dye is capable of being excited into that dipole state, so this state can decay and this one can be excited in a radiationless way. This is Foster resonance energy transfer, and then with the usual fluorescence process here, a photon is emitted at long wavelength. When the 2 dyes are very close to one another, this resonant energy transfer is very efficient, and the result is that if you illuminate the pair of dyes with blue light, purely red photons will come out. However, if you separate the 2 dyes, then one will see green photons coming out if you illuminate in the blue, because the resonant energy transfer is no longer there. The expression for the fret efficiency of a pair of dyes that do this is given here. Because it's a dipole-dipole interaction, the distance dependence is like 1 over the distance to the 6th power, and so when d equals this parameter r0, the efficiency is 50%, meaning that half the photons would come out in the green and half the photons would come out in the red. This Foster radius is something that can be calculated. However, there are all sorts of parameters that affect it and the way in which the dyes overlap with one another. So as an absolute spatial ruler, fret is still a questionable technique, but as a way of saying are two things near or far, it is fantastic. This slide here shows an illustration of the use of a fret. So this is a rather elaborate DNA structure. You can see a bit of double helix here, 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 and here. It's called a holiday junction, part of a process called recombination that we'll talk about in Chapter 10. And so you actually see that the helices cross over here and form this X-shaped structure. So here is a pair of dyes, here one emitting in the blue and here one emitting in the red. And so when these dyes are far apart, there is no fret. However, this structure is capable of flipping into this cruciform shape here where the dyes are quite close, and then flipping again into a shape in which the dyes are extremely close, allowing resonant energy transfer. So how long does the molecule spend in this state shown in panel C? Here is a set of fret signals, so you can see the red emission, which is characteristic when this thing is illuminated in the blue, of a fret signal, a resonant energy transfer, is high. It goes low. Notice the green signal now goes high. So these pairs of signals are anti-correlated as the molecule flips. By taking the ratio of these two signals, one can plot out a fret efficiency, and here it is as a function of time, varying from about 0.7 in this configuration to 0 in one of these configurations, and therefore one can analyze the dynamics of the molecule, and in particular, how much time it spends in this configuration. Notice, because you're taking a ratio of these signals, the signal to noise in this single molecule signal is remarkably high. Because of the need to integrate enough signal to get a single molecule signal, the time resolution is limited to perhaps about tens of milliseconds. However, these units along here are seconds, and you can see the molecule spends quite a lot of time in this configuration, which is why this experiment worked. What happens if one wants to look at faster processes? Well, there is a way of doing this, which is just to measure the distribution of times between fluorescently emitted photons. So if you look at a signal coming from an avalanche photodiode, you'll see spikes randomly occurring as a function of time owing to the emission of photons. Now, if the dye is diffusing very slowly through the collected optical volume of a microscope, then the interval between photons would be what you would expect in essence for a fixed point. If, however, the dye is diffusing very quickly through the focal point of a microscope, then the interval between light photon emissions increases. And so by looking at this correlation function, in other words, the fluorescence at some time t, multiplied by the fluorescence at some time t plus tau, and then integrated, one can detect, first of all, a correlation owing to the fluorescence lifetime itself, so an exponential decay, and then perhaps a slower exponential decay owing to diffusion of the molecule, and in some cases, internal modes of motion of the molecule.